demo and discussion on 3D scanner, 3D printer, and BioVia software. So today we have a resource person, Mr. Sridhar M, with us. Who is going to discuss and present whole these three instruments and as well software. So, Mr. Sridharan, welcome, and it is over to you.
Yep. Thank you, ma'am. Thank you, ma'am. Good afternoon, everyone. I'm Sridharan from Alchem Technologies, Bangalore. Our head office in Bangalore. We are a technology-based company working across like various latest emerging technologies like 3D printing, digitization technologies, and artificial intelligence, and so on and so forth. So now the topic is going to be, to start with, Mr. Lakshmikant Sharma will explain. Okay. So. Okay. So Mr. Lakshmikant Sharma will explain you through 3D scanning live demo, and its applications throughout the Artex Studio software. Then to Miss Aishwarya, Miss Aishwarya, she will explain you about bioprinting, 3D biosoft applications. Yes, over to Mr. Lakshmikant. Yeah. We'll start with the demo. Okay.
Good afternoon, everyone. So this is Lakshmikant Sharma. Thanks for Sridharan to give introduction about myself and our company. So just to tell about us, we are a complete pan-India company having offices in all over the India, operating with all the kind of industries and supporting for the almost all kind of defense or automotive suppliers as well as consumer products. So just
check once
is my voice is audible to everyone?
Yes, sir.
Hello. Yeah. Okay.
Yes, sir. It is
so
audible.
Just to go on, as I said. Yeah. Okay. Thank you. So, as per today's agenda, I will be taking care of all the 3D scanning activities. Just to speak about scanner side. So the company what we deal with for the 3D scanners are called from the company OEM is called Artec 3D, and again Artec 3D is a Luxembourg-based company, like it's a European made, and they are also in the industry from more than a decade, and they are having a very good market in US as well as in Europe. Extensively, some are using for the applications of forensic side. Just to tell about who all are very basic to the 3D scanners, just give a basic introduction about 3D scanning, what it is. 3D scanners are just like devices which are used to exactly get the digital copy of any of the physical components. Okay, so that means you have some physical component. Once after the 3D scanning is done, whatever the digital model you will get, it will be having all the details of the size, shape, as well as the it's like just like a 1:1 model of the exact physical copy. Okay, and then one more advantages of these Artex scanners are that these are also can be able to get the real color as well. Okay, so not only geometrical details as well as with the real color, which can adds more details to the new way of understanding the things as well as to the understanding the analysis side. So how these scanners work? So as I said, these are like non-contact type, because if you are applying these scanners in the applications of forensic and all, it has to be undisturbable, right? The situation should not be disturbable. So in that case, these scanners are non-contact type, and it's works on the principle of triangulation. That means projector and a camera based, and scanners will calculate the distance automatically from the scanner to the object, and it reconstruct the entire scenario virtually. Okay, so that's how these scanners works. And what's the need of 3D scanners basically? So as we know that 3D model is having all the details of XYZ coordinate, XYZ. So these are some of the general methods like how to get the 3D models in terms of physical world. And similarly, there are methods to get the 3D models in terms of design or computer-based orientation, like parametric modeling or sculpting. But how about if you need something which need to be integrated between physical world to the digital world? So there comes the application of 3D scanners. So that's what 3D scanners do. So if you have physical component, once you do the scanning, you will get the exact replica of the physical object. I hope this is clear. I will just move on to the next slide. Yeah. So the scanners, what we have, it has a capability to even to scan the as small as, like I can say, jewelry ring to as long as the entire architecture or the buildings, monuments, or even some are using for the city plannings for those kind of applications. Okay, so it all depends on basically 2 or 3 parameters. One I can say like the size of the component what we are scanning, and second is what detailing we are expecting from the object or else the thing, and third is the what application, end application. And these are some of the products. I'll just don't brief about all those things. I'll just directly move on just by telling one points for each scanner. So this particular Artec Micro is for generally scanning less than 10 centimeter per second, usually which will be helpful for scanning dentistry or jewelry or any kind of minute micro tools for those kind of applications. And similarly, the other model is Space Spider. Space Spider is ideal for scanning less than half meters apart, and usually it's and the scanner accuracy is almost 50 microns of accuracy, and detailing quality will be of 100 microns. So again, the other model is Artec EVA, and Artec EVA is again here the difference between EVA and Spider is that one is the lighting source, and the major thing is that the capturing area. Okay, in the Space Spider, assuming that the capturing area will be around 150 by 150, whereas in the EVA it will be near to half meter. Okay, so that means it's very easier to scan the larger components with a very less time. So the major advantage of Artec is that we are the only scanner which comes up with the external battery pack or the battery pack support. That means it can be easily usable for the outdoor applications clearly wherever there is no power supply. And the newest model is Artec Leo, which we have proposed.
Yes.
So the Artec Leo is the upgraded version of Artec EVA, I can say, in terms of battery. Like the battery is completely inbuilt, as well as in Artec EVA and Spider, you need to connect it to the laptop for the real-time scanning. Whereas in the Leo, everything has been inbuilt. Like it has a complete touchscreen display. We'll discuss it in the next slide. So again, the next model is Artec Ray. Artec Ray is a long-range scanner. As I said, it's for basically scanning more than 10 meter apart. And in one single shot, it takes almost 110 meter. Okay, so it's generally for city planning or to understand the if it's any natural calamities, the situations are there, or if you want to for the security reasons, you need to scan the entire buildings. So in that applications, it will be very useful. Okay. So just to tell about ourselves, I'll just play video as well as, and parallel I'll explain the tools. So this is the model Artec Leo, which is the only scanner in the industry which can be used completely 100% portable, like no external connection. Just like a video camera, you can just turn on the scanner, you can directly go to any location, you can scan it. And the battery pack gives almost 5 hours of backup. And the drawback was that till the previous scanners are that it could not be able to scan it in the direct under sunlight. But this scanner has the capability to scan in the direct sunlight as well. And it's the only scanner in the industry which is having a complete onboard processor with the complete HD built-in display. So these are our and these some of the quick facts what I have mentioned here. This will apply to all the scanners. Like our scanners are completely handheld, and some of the scanners are like less than 1 kg, as well as it comes up with a battery pack. So that's what makes it a more reliable, because if the applications like forensic and for the excavation applications, the major problem is that the battery means the electric supply is a very major problem. In that case, Artec 3D is best suited for that applications. And as I said, this also captures the real color as it is. Whereas the existed scanners in the market, you have to put a marker on the location or the marker on the object before doing the scanning. So what the drawback is that once after the scanning, you will get the data of the markers as well in the final output, real digitization. So in our case, it's not needed. So we can digitize the exact copy of any of the physical component. And lab temperature is not required. That means these scanners can be operable within almost 55 degrees Celsius of range. So it's highly sufficient for any kind of, whether it's a hill area, whether it's a, what I can say, in the mines or anywhere, it can be easily usable. And one more thing, the major advantage is on the software side, which we call Artec Studio. So Artec Studio is so user-centric pilot mode, which directly converts the raw scan data to the final polygon model. Hope this is clear. Just directly skip on to the applications where it can be used in the forensic applications. So one is, I can say, forensic topology. So the major, as we know that nowadays the making a documentation is the major problem. Okay, in terms of like assuming if some of the crime scene has happened and taking the photographically, even though if it's a high-end cameras, taking the photographs and it's studying those photos are a bit difficult task, correct? So how about if you can scan the entire scenario as it is, and you can sit in a virtual room and you can clearly make clear detail3D scanning the footprint of the culprit, and just for the identification purposes, whether the footprint belongs to the same person or not. And similarly, unlock the get the details of whatever belongs to them. So for that applications also, these 3D scanners can be usable. So again, as I said, for the forensic anthropology, basically exactly like after the documentation, the entire crime scene will be deleted or it will be removed. Shall I turn on the video?
Yes, yes. Hello. Hello.
Hoping video is visible now. Is it visible?
Yes, sir.
Yeah, okay. How about the presentation now, ma'am? Its presentation is clear. Yeah, okay.
Sir.
And one more application is that whenever they get the skull or anything, even though there might be a lot of theories around it, right? Every case will have its own stories. In that case, even to understand exactly what's the trauma behind that, even before the death or after the death, it can be clearly visualized with the 3D scanners. And one example is that for sometimes whenever the crime scene is detected, at exactly that time, there might be chances of there is a lot of natural facts will be affecting that particular scenario, right? So that minute details might not be captured in the by photos or just by virtue of the person who is in that crime scene. In that case, if the entire scene is clearly 3D scanned, even they can clearly understand what exactly the natural phenomena has been applied on the happened on the forensic, exactly the crime scene. And sometimes, as I said, like some animals might have taken the part somewhere and they have been shifted to some distance, or some of the even the criminal or the person who might be committed the crime can also has not put some of the evidence in and around the surrounding. In that case, exactly same kind of scenarios can be captured and it can be clearly studied. Okay, so again, this is one of the application, one of the forensic lab they are using for the study. Exactly the same way what I said, they will, whatever the bones they are getting, they will clearly collaborate and they will try to match it with the master template or what they call digital encyclopedia. So with the bone encyclopedia, they will match it, and based on that, exactly going by detail, whatever the things are missing, and in reference to the remaining bones or remaining details, they will check what are the adjustment ratio between those two, that so that they can have a clear understanding of it, as well as they can digitize it clearly. One more application is for the cadaver dissection. It might be educational, but again, this is the most needed thing in the current situation. Why? Because getting the cadavers are very difficult in the industry, as well as sometimes to teach the people how to do the dissection, or some kind of virtual training, I can say. Instead of directly operating on the crime scene or directly operating on the how to collect the data and all, you can virtually conduct the entire scenario first, and then based on that, we can make the persons to be on the spot and to get the 100% result. And one more thing, this is a very trendy thing in the current forensic department industry, is that wherever the digital facial construction. So this is one of the lab in UK. What they are doing is that wherever they get the, what I can say, the skulls or anything, it might be they will reconstruct virtually, taking the skull scan as a reference, because from the DNA and other things, you will get the detailing of the how exactly the person might look like, what's the hair color and other details. But how to recreate that, you need some exact reference of the skull or the which belongs to the actual person, correct? So in that case, 3D scanning plays a very major role, and only possible application, only possible way to the getting that exact detail of the skull or the object, and then taking that as a reference, they will build the skull, and digitally they can bring back to life, even though person has been died so long. And the same application can be applied to wherever they get some of the what I can say, unnamed or unfound dead bodies. They can recreate, and they can make the exact they can make it to make publish, okay, this is the person to whom we got the body, or how it has to be taken care. Yeah. So other than that, it can be very much usable in the applications like traffic collisions, as well as train, plane and transportation causes, as well as these are like not exactly with the Leo or anything. Even if it's a room kind of scenario, so just only room based, or if the thing is not more than 7 to 8 meters, it can be done with Leo itself, but having a high-end computer system in the backend for the processing side. So other than that, if the crime scene is more than some 10 meters to 20 meters, we can use the applications of like the long-range scanners, as I showed in terms of Arctic Array. It can be clearly calculated, and it can be used. These nowadays, even the forensic departments are using these 3D scan data for the courtroom application, just to show to the judges or to them how exactly the crime scene has been created. Virtually, they will create, and they will do the animations as well for the exactly how the things has been happened, and they can submit to the courtroom applications with the exact real scenarios. So now, assuming that the scan model is ready, after that there comes the application of from the scan model, whatever we get, usually it will be in the form of it just like a triangulated format, okay, just like a mesh model. So some applications requires converting that to the CAD model, okay. That means when we want to take this output from the scanners to different softwares which will support, if the software is supporting directly the polygon models, directly you can take the output of the scanner. And if it is not, in that case, some softwares like design softwares might require supporting formats, right? In that case, we need the softwares like reverse engineering, where you can import the scan models, we can work on that, and we can convert that to the CAD models. Yeah. So what the one we suggested to our customers, or one we deal with as a package along with the 3D scanners, are the 3D experience platform, which is from the Dassault Systems. And again, it's completely customizable, design, manufacturing supported software. We can even customize the even if the multiple people are operating on one design, they can collaboratively work together to get exact pinning and what is the status of each project. So these are some of the industries who are these Dassault System products. And if you want to add any points, you can add. So again, this is
The workflow.
This is about the workflow, how that it will happen. Like from the scan data we'll get, if you want to convert, as I said, for the any digitized based design site, so we can convert into the digital, optimize that, even the design as well. And basically this is for analysis purposes, I can say. If you want to customize or optimize any of the designs, we can do that. And then we can 3D print it in a software. And this 3D printing is completely different. We'll talk it in the next slide, what exactly these 3D printers are now. And
yeah, this is we have two technologies in 3D printing. One is FDM, fused deposit modeling. Second is PolyJet, multicolor multi-material 3D printing. This slide is exactly to show how the workflow goes. Like from the inputs are two actually. One is 3D scan data. Second is own design. We do our own design and get it done. Then we bring it to the 3D printers software. That is what the 3D printing software makes, and we have an operational software for rapid prototyping shop. Then yeah, that can be brought into either the FDM machine or PolyJet machine. The first one we call it as FDM machine, and second one we call it as PolyJet machine. Okay, so then post processing, based on the required post processing methods, we can make it. We will show you some of these samples, what this makes up to, how this technology works. FDM basically works on a it's completely plastic-based 3D printer, and works based on a heated material flowing in a particular pattern designed by the software. Okay. Then PolyJet, it's completely resin-based. The earlier one, FDM, is spool-based, as a plastic, rigid plastic, filament-based, and heated into a particular pattern and flown through. The second one is completely in resin-based. It's been treated once it has been laid down in the desired shape. It's been treated by UV light, and the shape forms. Yeah, these are all some of. Yeah, what happened? Okay. Yeah. So these are all some of the sample printed in healthcare as well, forensic analysis. Based on the required virtual history and all, it has been created. So
okay.
Yeah.
So just like in the virtual autopsy, what they can do is that even before actually the just by taking the CT scan of the person, meet exactly what has happened inside. They can create clearly make the digital copies of the 3D printers. As you can see this, even we can make the components like exactly transparent. Or even if it's any internal dents or internal marks are available, or anything has happened to the person when he died, based on the density we can segregate, and based on that we can 3D print, and we can have a clear understanding how exactly that has happened, even as it's a proof of concept. Okay. And similarly, one application is that from MRI CT scan we can get that scan model. And then one more is that even if it's for physical analysis, like as I said, even the skulls have been 3D print, the skulls has been scanned, and just by keep on studying on that, it's a bit difficult. And if you want to have the physical feel of that particular skull or any of the parts belong to that, you can clearly take it out, and we can 3D print with any of our technology, and you can have a clear understanding how exactly that has been happened and what else has been happened to that. Yeah. So that's it. So this is all about 3D scanners as well as 3D printers, along with the Dassault System 3D Experience platform for the design analysis. And
any questions
as of now
on this topic?
No, sir. Not yet.
Okay.
I'll just stop my share screening just for a minute.
We will show you live demo on this. Show it. Show the screen.
Yeah, just cast the screen.
No, show it. How to cast the should know. We are just connecting it to.
If you can connect in mobile, we can show this. Otherwise, it's Just a second. I have a clear understanding how fast these scanners can perform.
What happened? Internet issue.
Yeah. Yes. Shall we proceed with the presentation parallelly? We can do it.
Yes, do that.
Just a second. Sorry for the delay.
Internet will be disconnected from there.
Are we audible? Hello?
Yes, sir.
Yeah, okay. Just a minute, man. Just a minute. Just trying to figure it out. There is some issue with for the casting the screen of the scanner.
Because of internet, not because of.
Have some Wi-Fi issue. Yes. Is it possible to connect from mobile?
Yeah,
Yes.
yeah.
We just connect. We just show it in mobile. Video. We just let it showing from here.
Yeah. Okay.
Okay.
Ma'am, can you look at the screen? Are you able to see the screen? Mute the mic.
Just speak. Just
Yeah, mic is on. Mic is on.
yeah.
Can you see the screen, ma'am? My mobile share. Hello.
Ma'am, are we audible?
Yes, sir, you're audible.
So this is the Artec Leo. So this is the Artec Leo. As you can see, this it's completely onboard processor scanner. There is no any connections separate in between. So as you can see it here, this comes up with a one battery, which gives almost 6 hours of backup nearly. And this is the touchscreen display, and it's a tiltable touchscreen display. It's not showing it. Just ask them once. Please.
Ma'am, are you able to see the screen?
No, sir, it is not visible.
Mobile share?
Uh,
Not visible.
Mobile share is not visible, sir.
Mobile share is not visible. Okay. Just a minute. I will check.
Just a minute, ma'am, please.
Is this connected to Altentech or something else? See, this is
Yeah,
connected to
that's
Altentech. You have to connect it to LK Sharma. Then only it will. Then it will work. Yeah. Only then. share the screen. Share the screen. Entire screen. Yes. I'll remove it.
Video you just shoot.
Okay.
Yeah, now my screen is completely visible now. So this is the scanner. As I said, you can see it here. It's a completely HD display, complete HD display where you can directly start scanning. And this is the camera, as well as this is the light source for the projector lights, I can say, as well as it has its own lighting capacities, what we call textured light. That means it also captures the color details for the assisting the color details, irrespective of the lighting condition. I'm just starting to scan now. Yeah. So as you can see it here, you can turn on the video.
Yes, it's been turned on. It's been turned on.
Possible, we'll just show it. So as you can see here, the speed of this scanner is almost 80 frames per second. So that means in one single second, it takes 80 different multiple scans. Okay. Just ask if my screen is visible.
Yes, zoom here.
But not about that. I have shared one slide also
Okay.
about that window also.
You present it.
Yeah. Okay. So just like this, you have to focus, and its working area is almost half meter to 1 meter. You just click on the button, it will directly start scanning. Just like this, we'll get the real scanning, real color. Yes. And as you might be observing that it is just like a real-time scanning. It's just like taking the video. If I need some other areas which is not capturing, I just have to focus it from that particular orientation, so that I'll get the complete scan data. And right now I'm scanning Miss Aishwarya, but again, I'm not getting any details of her back of the face. Why it is because I'm just only focusing from the front side. And again, for the neck areas, I'm just focusing from here. Similarly, as you can see it here, it's a real-time constructing everything. If I need something from the back side, I just need to focus it from the back end, and it will clearly matches to the whatever has been captured on the front side to the back side. So you don't need to worry about it. Just like just walking around the object, and it will be reconstructing everything in real time. Yeah. So that's it. You can see here. If you are seeing my screen, I can show. Maybe my screen is presenting, but however, I'll try to show it here as well. You can have a clear look of that, how exactly it is looking like. But still, it's a raw scan data. The next process involves just directly transfer this data from scanner to the laptop, because the scanner has its own capturing data as well as a storage capacity of 500 GB SSD, which is highly sufficient for scanning any of the larger components, even the entire crime scene completely. So after this scan model, I'll take this scan data to the laptop, either through Ethernet cable or through Wi-Fi or any other option like copying to the SD card. And then we'll do the post-processing in the Artex Studio 15 software. So parallelly, I guess, how we do close it out.
Within minutes, we'll show you the processing. Ma'am, you saw entire process? Can you
Yes,
hear me?
sir. Yes, sir. Yes, sir.
It is on. It is on.
My video is off. Yes, it's
Who is in there?
No. I left the meeting.
Can you see there?
Just a second, ma'am. Actually, we turned off the speaker in. Yeah. So, is the scanning screen was visible to you, ma'am, when we are doing the real-time scanning?
It's a mobile screen. Is this?
No, it's a computer screen, ma'am. Computer screen.
Yes, sir. It's visible.
Okay.
You saw the process, how we scanned her, and
It's hardly 1 or 1.5 minutes. That's it.
So the scanning procedure was like not yet, means not that clear, but yes, we were able to see your desktop screen than your mobile screen.
Okay. Then it was clear, ma'am. Like that, actually, you might have observing there was something showing in green color as well as in red color when scanning was happening. So that's a very unique feature of Artec. What they have given is that a quality mode. That means when you are scanning, whichever the area is not showing in red color, you just focus it some more time so that till it turns to green. That means it automatically understands whether the data is capturing is sufficient for the post processing or not. Okay, so based on that, you can hold it for some more time, and once it turns green, you can just move it to the next location. It's just like matching between one single photo shot to the next photo shot. And similarly, it takes 80 80 different 3D shots in 1 second, and it will keeps matching as you are moving around the object. Like that, it will be recreated. I guess still you are able to see the screen, ma'am, whatever I have shared.
Yes, sir.
Yeah, you can see it here. You can see it here clearly. And based on that, if it's any areas you've been missed out or anything, you can again start rescanning. And in Artec Leo, it will be starts rematching to the same location from where you have left out. So you don't need to be bothered about any of the location, even if you stop the scanning. Again, you can start it from anywhere. It will directly starts matching to the same location from where you have left, and you can finish the scanning. And combining this and to the scanner processing capability, we claim ourselves as one of the fastest scanner in the market. Why? Because it doesn't require any of the markers and other calibration. Like with the other scanners, we have to do initial calibration before start doing the scanning. That will only take almost like 5 to 10 minutes or minimum. Whereas within that time, we will finish off our entire scanning procedure. So that's why it's called one of the fastest scanner in the market. Yeah. So this is about it.
Yeah, this is about 3D scanning and its applications on forensic department. We would like to continue with Ms. Aishwarya's presentation on bioprinting, 3D bioprinting.
Before that,
Any questions, ma'am?
Yeah, please. If you have any questions or any detail required, please let us know.
So we'll ask questions after the completion of session.
Sure, ma'am. Sure, ma'am. I'll ask Ms. Aishwarya to start immediately. And ma'am, can you accept Ms. Divya to participate in the meeting? She's still waiting.
She's one of our colleague and
presenter on this for BioVia.
Excuse me, Bridges, sir, can you please let her in?
Yes. I'll just ask her to. Aishwarya, you can start. சரி கொஞ்சம் ஃபுல்லாக சைன் அப் பண்ணிட்டு மறுபடியும் லாகின் பண்ணி ஒரு 15 minutes இருக்கு டைம் ஆகும் பட் நீங்கள் லாகின் பண்ணுங்க.
Hope the screen is visible to everyone.
Yes.
It's stating Cellink Life Sciences.
Okay.
Is it visible, ma'am?
Just a second, sir. It's not visible yet.
Oh, okay. Okay, I'll just reshare it then one more time. Yeah, how about now, ma'am?
Yes, sir. It's visible.
Yeah, okay. I'm handing over the session to Ms. Aishwarya. This video is not on. Please turn it on.
Yeah. Hi, I'm Aishwarya. I'm the technical specialist here for a product called the bioprinter. So,
You are visible.
am I visible?
Yes, ma'am.
Yes, all right. So I just begin with this. My main area would be the 3D bioprinter, and as you know that we have a couple of products out. For example, the scanner, the 3D printers, etc. Similarly, you also have a bioprinter, which is from the company called Cellink. So Cellink is one of our representative companies, and they have quite a lot of products in the area of say biological sciences, biomedical, and biotechnology. This machine is something that can benefit something in most broad applied sciences sections of biology. So starting with the machine itself, right? Starting with the machine, or rather the technology, I would talk about what 3D bioprinting is. So 3D bioprinting is a form of additive manufacturing. So additive manufacturing would mean a layer by layer addition of layers, basically, to create a large structure. So in this case, we're actually using cells and other biocompatible materials. These biocompatible materials that you have are called as inks or bioinks, and they are meant to basically recreate any particular structure of the body. So reminding you about the application, it is something mostly in the field of tissue engineering, or say example cell cultures, etc. So here you are taking the cells and you're trying to mimic the behavior of natural living systems. So I just show you something very simple here. So basically you have something called a biomaterial mixed with your cells, which are your main components, and they're printed in the form of additive manufacturing in order to create a structure or a design that you want to create for an application. So 3D bioprinting itself was something meant for a specific application, but now we're in this phase wherein we are going step by step. So I'll just play a small video so that things are more clear for you.
There are currently hundreds of thousands of people on transport.
Are you able to see the video and hear it as well?
Yes.
Transplant lists waiting for critical organs like kidneys, hearts, and livers that could save their lives. Unfortunately, there aren't nearly enough donor organs available to fill that demand. What if, instead of waiting, we could create brand new customized organs from scratch? That's the idea behind bioprinting, a branch of regenerative medicine currently under development. We're not able to print complex organs just yet, but simpler tissues, including blood vessels and tubes responsible for nutrient and waste exchange, are already in our grasp. Bioprinting is a biological cousin of 3D printing, a technique that deposits layers of material on top of each other to construct a three-dimensional object, one slice at a time. Instead of starting with metal, plastic, or ceramic, a 3D printer for organs and tissues uses bioink, a printable material that contains living cells. The bulk of many bioinks are water-rich molecules called hydrogels. Mixed into those are millions of living cells, as well as various chemicals that encourage cells to communicate and grow. Some bioinks include a single type of cell, while others combine several different kinds to produce more complex structures. Let's say you want to print a meniscus, which is a piece of cartilage in the knee that keeps the shin bone and thigh bone from grinding against each other. It's made up of cells called chondrocytes, and you'll need a healthy supply of them for your bioink. These cells can come from donors whose cell lines are replicated in a lab, or they might originate from a patient's own tissue to create a personalized meniscus less likely to be rejected by their body.
So this technology, which is 3D bioprinting, is fairly a new technology, but there has been acceptable amount of cases that will actually prove how effective or efficient it is, or how close we are to creating more artificial organs or artificial systems. Now, from the basic understanding what 3D bioprinting is, we have machines, or we have devices or tools that work in different principles. So we majorly deal with machines which are by two methods. One is extrusion printing, and another is called digital light processing. Extrusion printing is basically pressure assisted. So use pressure pumps or something. There is a piston or a cartridge. This is where the pressure is exerted, and you have your biomaterial or your bioink extruded to create a specific model. Right? So here it is filament dependent. And here you can create multiple models and use many types of materials. So I will get a little more into detail of the materials later. So just another quick video to tell you what extrusion printing is. So as you can see, it's just based on a Petri plate where they're printing something. So this is just a basic model. Obviously, there are better things that can be done out of this. Next is another technology or another principle here, which is called the digital light processing. So the way it works is on the dependence of light. So light is used, and here you will have layer by layer curing. Curing basically is cross-linking of materials in order to form a structure. So it's meant to have a very high resolution, and it's supposed to be watertight. When you say watertight, you're trying to create more fine structures, more intricate structures, and you also have a specific set of materials only which follow the principle. Now another video on one of the what do you say, the working of it, or how it actually seems to you. So this is basically a build platform, as you see, and this is one structure which just gets printed. So it gets printed layer by layer upside down. It's meant to have a really good resolution, and yeah, as you can see in that image, they create a very perfect module. So this is the types of printers. Following this is the steps involved. Like our 3D scanners or our 3D printers, this also has a set of steps which it follows. So you start with, say, pre-processing. Pre-processing is example, I have an application for skin, right? So for skin, I will select, say, my skin cells, that is keratinocytes, fibroblasts, melanocytes. Then I also have selection of materials, biomaterials or bioinks. These can be both synthetic or natural, and also the CAD model. CAD model is basically what morphological design you want to show. Now, say I want to print something like skin, it is more like a flat structure. Although it is something in 3D, we're trying to create a 3D background for the cells, but this is something which follows a particular pattern which follows our physiology. So the main idea is to follow the physiology and provide an environment which is more three-dimensional, right? So from pre-processing, we go to processing, and processing is like what technique you're using. You're, say, using extrusion-based printing or light-based printing. So there are a lot of factors to consider there when you're printing. Say, example, the print time, print speed, the temperature. So these are all essential factors to consider because you'll be using cells. So after that, you have something called post-processing. Post-processing is differentiation of cells. You have the incubation of cells. You allow the growth of the cells, and you also see how they function from time to time. So as you grow a tissue or as you grow a particular cell line, you have to also evaluate it. Also, you have to check whether it is producing the biomaterials it's supposed to produce. Also, it is looking healthy, and it is doing the functions it's supposed to do as an organ. So we are trying to get closer to creating a whole organ starting from tissue level. So when you start with a tissue level, you are able to see whether this can progress further or not to creating something bigger. So obviously, once the steps are done, you have it to be used for the applications. And now another point to add, 3D printing, 3D bioprinting, what you would say, is something where you don't have to just use any type of cell. Like you don't have to be like, okay, I have to use a particular cell line from somewhere. This is more customizable. Say I want to do it for a specific patient. I have an application for one particular patient. I want to recreate something for that patient. Then I can actually use that person's cells and mix it with these biomaterials and create a model for that person. I mean, this is just for the case of immunology aspects, because we don't want any sort of rejection or anything. But otherwise, people for testing purposes always go through standard, cell lines or whatever we have to use. Now, the applications of bioprinting, I mean, this is something which is quite required in all aspects. So you have tissue model. Say now for tissue model, you would go for trying to create a replica of a particular tissue, or you're trying to create something which is a solution to an issue. For example, I have a nerve issue, a nerve degeneration at one point. So you want to create something that can be a junction between maybe two nerve ends. So when you use these tissue models, example, I'm using a nerve tissue model, I'm trying to create something which is to accommodate in that sector of the body. Similarly, you have next thing, which is organ on a chip. Organ on a chip is a platform wherein you're putting multiple organs, multiple tissue types in one platform in order to carry out maybe testing purposes, and it requires a very important application of most important field, which is microfluidics. So it is a very diverse, it's got a very diverse set of applications here. Similarly, there comes 3D cell culture. So we know that we are all 3D structures, and yet we are still going with 2D culturing, right? Whatever test you do, toxicology testing or drug testing or even any other testing that is happening right now in the field of medicine or even forensic sciences, probably is happening only to the 2D level. So we are also trying to like create this more 3D structure for the cells. You have biomaterial development, something which is more relevant to printing as to creating materials for the printing. Drug delivery is another application where you're creating models that allow in the drug delivery. You're creating a system, a smart system that allows a drug delivery. Followed by soft robotics. Again, this is a fairly new field wherein you are trying to mimic parts of the body to a more soft extent. So coming to the applications in forensic sciences, one thing is, if you want to go to the area of forensic medicine and you want to do more studies, you have accident and wound regeneration models. So now this is one case wherein you have the regeneration of critical size mandibular defect. So here you use a 3D bioprinting scaffold, and this is supposed to do a guided bone regeneration. So you're creating a bone module. So example, there is a person who's had a mandibular defect. There's a defect in his lower jaw, and we are trying to first thing understand the extent of the issue. So you probably use a 3D scanner, followed by putting in this application where you're using a 3D bioprinter to replace that particular part, right? So you use that defect and you try to create something in place of that. So here you've used maybe two materials, hydroxyapatite and PCL, and also mesenchymal stem cells. So basically a type of stem cell. And you're trying to replace that particular part which is defective with something artificial and trying to grow that part. So here, by results, we saw that after 4 weeks, there was mineralization and secretion of collagen material. So basically, these are essential components for the healing of something. Following that, you have skin grafts and wound healing. Then you have accident, impact test, and disease model. Disease model is something that can really, really benefit, considering how so many medications can affect so many parts of your body. I'll get in detail there. So following this, you have a very basic platform. It's creating a 3D cell culture platform for the cells to grow in. So we know that, like I said, we are 3D structures. So we are seeing, if you see the images here, you will see that there is a comparison between 2D on top versus 3D in the bottom. So you'll see how efficiently there is a growth, how we are trying to replicate something so perfectly that it shows that environment well. Now, also giving you a little bit benefit, say, example, there are cells. There is a requirement of cell-cell interaction. There is a cell matrix interaction. Cell matrix is in cell with the extracellular matrix. Then you have diffusion kinetic studies. You have in vivo tissue similarity, because we are trying to test something on something similar to the human body. And there's also mechanical properties we need to check. So the other major application where it can really be working on is in the toxicology department, where you have, say, high throughput screening of drugs or drug discovery or testing drugs. So this will create a platform where you're checking the metabolism, pharmacokinetics in the toxicology majorly. So there's a case wherein they try to understand the impact of a particular drug on a healthy person or a healthy cell versus an unhealthy person or an unhealthy cell, right? We're trying to create macro structures to more miniature versions to try to interpolate and then understand what is going to happen out of it, right? So here they've used, say, example, hepatocyte cells, right, liver cells, and now they've used a drug which is trovafloxacin, and now they're trying to check with so many doses, so many length of doses, how will it affect your target organ. Now, in this way, you can try it on multiple types of cells and see how a particular medicine, how a poison, how a drug, how anything can actually affect that part of the body, so that you can interpolate and see, okay, something similar to it or the same thing, how it can actually affect somebody to harm or maybe disarm the person, right? So there are results. I mean, there are some good results where they try to test it, and they were trying to correlate and match between similar things so that they could avoid or even understand what is the cause for a person's death, or say create that scenario for more studies or more research. So now we are progressing from pharmaceutical and diagnostic development. So we are going from drug discovery, where you're going from simple printed 3D models. Then you go to complex disease models. So, say example, somebody has a heart attack. You're trying to, see the impact of a particular drug on his heart, or a heart model, to understand how one particular poison or one toxic thing can affect the rest of your body. Right, so this is quite interesting in terms of research. Next is toxicity screening, as I told you, and there is precision medicine. That is when it is specific to a person. So this is a broader range of how the applications come in. And respective to that, we have our products, which are, say, Incredible Plus, BioX, BioX6, and Lumenix. So I'll give you a very, very light intro to the products. One is Incredible Plus. It consists of 2 print heads. So it's a basic thing. You can put 2 types of material, say, print simultaneously. It has this air chamber or clean chamber technology. It has UV cross-linking systems. For example, you have a biomaterial put in, you have the cells put in. You want that material to solidify a little bit, you need cross-linking. Then you have pressure regulators. Like I told you, it's a pressure-assisted device. You try to use that and put the whole platform. Then you have temperature controls, you have LCD display and pressure controls and few other things. Following that is something called BioX or BioX6, which is a slightly more, fairly more advanced system, which is a clean chamber technology to maintain sterility. There is again print heads, temperature controls when you're printing with different materials. You have inbuilt compressors. You have wide range of materials to be used. Then you have multiple types of print heads that you can swap. So for each application, each type of material, you can use each thing, and this is meant to basically replicate something that comes closer to your application. So we have large varieties, say alginate, gelatin, collagen. Then you have thermoplastic materials, photo inks. All of these can be used in this machine or any other machines that we have. Say both Incredible and BioX systems. Lumenix is a fairly more precise machine, which gives you a good resolution, a better biocompatibility, and major thing about this is it provides a very good geometry. It enhances the vasculature and the porosity of any scaffold or any structure you're trying to create, and it has good light intensity distribution, or something that you can control easily, and dimensions as small as 150 micrometers can be created. So I mean, that's the marvel of this machine. Working principle again is quite basic. You have slicing, right? You have a 3D model that you've constructed. You take that, you slice it, and you try to regenerate it again. Again, materials are, say, PEGDA series material, GELMA series materials. These are something which we can get in detail in case you have any questions. And there are some basic case models. You'll be able to see how you're creating something this small, something so intricate, and we're able to actually visualize parts of the body or any type of structure easily. So with this, I end the session. Next, I'll hand it over to Divya. I'll just wait for her to start. Just a second.
Joined.
She joined?
Yeah.
I'll stop sharing.
Shivani ma'am, can you allow Divya to present?
Yes, sir. Actually, I am there.
Okay, right.
Yeah, I am here. No problem. You all can see my screen?
Yeah, we are seeing.
We're able
Yes, yes, thank you so much. So, very good afternoon to everyone. It's a continuation of what Aishwarya has been speaking. So, this is a forensic application of a BioVR Discovery Studio. This software is basically an in silico computational biology and chemistry as a solution for various application. So one of the application is a forensic application in a pharmacology, or you want to do some kind of docking analysis or structure-based analysis. This tool will be more feasible in working on different perspectives of the discovering and then research. Let us walk along and see about how this tool will be helpful for you in forensic science and pharmacologies. Before that, I would like to introduce about the BioVR Discovery Studio. It is a rich scientific portfolio. We have it from unified lab management, where we are trying to replace the paperwork into an electronic laboratory notebook, and of course, moving into the process production and operations. And we are also into the quality and regulatory management, where we try to process documentations, compliance, and collaborations. And if you expand more into the collaborative sciences, right from the predictive model, if you have a scratch of a sequence or a compound, you can able to end up with a lot of solutions for that. Maybe if you start with one particular compound, you can draw the conclusion for that similar like compounds and its effectiveness or its activity, or you can come to know about whether this particular molecule will be toxic or non-toxic. Up to toxic means up to which level, what is that measurement. So like kind of a quantitative and qualitative analysis can be done by using in a collaborative science. And more into, some people want to identify about the therapeutic of molecules like a macromolecules, that also can be done. And in the engineering materials, where we can introduce even the metal particles, we want to study more about the metals and its efficiency or its reactants. That kind of an analysis that can be done by using a material studio, using engineering materials. And chemicals and formulation, if you like to introduce some formulation for certain formulations of A and B, that also can be done by using in a BioVR Discovery Studio software. And if I expand about a collaborative science, it is, I already told, it is a comprehensive tool where you can analyze for various management with flexibility for an enterprise integrations. And if you take about a life science predictive analysis, it could be applicable for all the domains. It is not only limited within a life sciences, where you can include mathematics, biology, physics, chemistry, pharmacology, whatever the people where we can see the perspective of proteins and then ligands. This particular collaborative science projects can be applicable in BioVR Discovery Studio. And of course, if you want to explore more on material science, you can more into the material science predictive analysis. And in case if you want to design your own pipeline for your execution of a process, that also can be done. And more into the collaboration, externalization, cloud computing application, research applications, and chemical inventory management, bioactivity, chemical sourcing content, all are the part of the software which can be expanded, and you can also see in your different perspective and application of these tools. Moving into the life science predictive analysis, which is a major domain for us, where we try to introduce a biological modeling and simulation to understand more about how the real-time environment and motion of your particle when it acts in a in vivo system. We cannot see how an in vivo system has been operating, but however, you can come to know about by utilizing a modeling and simulation tools. And this is even applicable for small molecules and then simulation. And we have a tools to analyze the quantitative structure active relationship and also the toxic relationship. All will come under this particular category. As I already told, it is a comprehensive tool, and then collaborative modeling and simulation can be done for various applications of life sciences discovery research. Moving into discovery and development, say for instance, if you come to the pharmacological, we would like to know about first of all the disease. What is the major cause for the disease to occur? And if you expand and you look into a funneling of this disease, there are lots of proteins which is playing an integrated way in a network or metabolic pathway is playing a major role. So to identify that particular disease or in a protein or a specific genetic measure which is causing the disease can be easily identified and can be validated by utilizing a tool. There are certain tools and the databases like in a protein data bank or a protein data bank from like this. There are lots of databases are there which will help us to identify the disease causing by a particular microorganisms or in a particular disease like a cancers. You can bring out that particular disease and you can expand and you can look into a specific protein which is playing a major role, and then you can go for other kinds of analysis. That is an idea of an identifying disease. For instance, if I want to work out the same in vivo technology or in vitro technology, that will almost consume about 2 to 5 years to identify the protein that is involving in a particular disease. But here, it will not even a 1 week. You can come to and draw the conclusion that this is a drug target, which is playing a major role in causing the disease. See, for instance, there is one particular protein which is playing a crucial role and interaction. Because of that particular protein only the disease is occurring. That can be easily identified to network biology or system biological approaches. So target identification, it is not even 2 to 5 years. It will takes maximum, if you want to work it out, not more than a month to identify the disease and the disease causing protein. Once you identify that, then we have to come to know about the compounds. There will be a series of multiple compounds, and each compound has a different behavior and metabolic process that we can identify by introducing into a QSTR technique, like a quantity structure toxic relationship technique, and also you can come to know about the activity technique using a QSTR methods. So that will be go for a preclinical testing. Before going for preclinical testing, you can test your drugs for various methods that is available in Discovery Studio. We have an FDA approved model and national toxicity prediction model to identify the exact scaffold which is causing for toxicity or which is responsible for the activity that can be identified. So this kinds of an analysis, say if I take it for 1 to 3 years, it will not even take about a month to finish. So obviously we are utilizing only in the terms of months, not in terms of years. So now you come to know about which molecule is that you want to test, whether you can go for in vivo system. So finally they go for in vivo system. Before that we have a dose ranges will be there. Say for instance, I want to treat this mice with particular level of LD50 value, LC50 value. Those values I can able to get it by using machine learning algorithms that is based in Discovery Studio. So once I identify and treat it, that becomes very easy to my process. Then moves on into a formulation technique, and then moving further into a phase 1, phase 2, and phase 3 clinical trials and an FDA. So this is almost, it will take 16 years for a particular drug to be entered into the market to discover and development. However, when we try to use this kinds of computational biology and chemistry tools, it will reduce almost 3 to 4 folds of your time and increases and accelerate your drug discovery process. This is an overview about your discovering and development. Moving on into a high throughput screening, because we always prefer to go for high throughput screening. This is not an automated process and to identify an active process. See, if you have a lack of molecules, high throughput screening is not always encouraged, because every time you have to introduce lots of chemicals and then formulation that need to be done to screen to identify in active molecules. It is like if you have a pool of 1 lakh of molecules, picking the one molecule is really a hard task. So to understand, we know about the outcome will help us to understand the interactions, but it will take a long time. And moreover, it will be expensive and time consuming. So 1 lakh of molecules, it's not an easy process to identify it. And it can be automated, but it still need a lot of human interventions. But not all the molecules, because when you screen 1 lakh of molecules, not all the molecules cannot be tested for assay. So in order to overcome or to be an alternative for this technique, we have a method called virtual screening technique. This is such an easy method where you can introduce a large number of molecules. See, you have millions of molecules which have a different behavior, that can be identified and can be screened in such a way using a funneling technique. We call it a funneling technique, where we try to identify its absorption, distribution, pharmacokinetics, and pharmacodynamic properties, and then whether the properties, physiochemical properties, are really suiting for this particular drug, what is its mechanism, how it is behaving, all these things that we can able to identify, and we can come to know about the hit targets. So even though it is not only applicable for an inbuilt databases, you can perform even for corporate libraries or a virtual library that you're trying to create. And this mode of interaction will increasing your important tool for your lead discovering process, and it will be very quick and economic. So even if I test 1 million of molecules, I'm not going to end up with an uh, when you steal 1 lakh of molecules or 1 million of molecules, of course we are going to end up with a novel therapeutic molecules. That molecules can be taken for in a further high throughput screening. Rather than screening for a 1 lakh of molecules, it comes into about 10 molecules of screening in a high throughput method. Even moreover, it can be automated once you design a pipeline and you set your process. It can be automated and will be more cost effective and saves you valuable time during a research. That's why we are initially going to work with the virtual screening technique. After that, we are moving for high throughput screening methods. So we should know about what is significance. The significance of virtual scanning can be used to validate the active state of modeling and to help us to distinguish between the mechanism of the ligands, whether it is going to attack the molecule or like an antagonist, or inducing a system we call as an agonist. And it will help us to ease your drug designing process. Even though if you don't know the target, you can come to know about the target of unknown molecules or unknown drug targets. Also, you can identify and you can validate, and that can be converted into potential drug targets. So screening of a large number of molecules, and also you can identify the good affinity. If you have a 50 molecules in your hand, you want to identify which will have a good affinity for the specific assay or receptor. That can be easily identified, and this way of identification method will ease your process. So certainly there will be little bit pitfalls will be in a high throughput screening method, which can be classified into 4 major categories. So erroneous and assumptions and expectations, and then data design and content. So how much error it is, certainty or uncertainty is going to occur. We are going to test even that. And we can data designing and content. That is very important when you come to the virtual screening technique. Choice of the software, which software we are going to use, whether it has been yearly validated or it has been keep on revising with the different algorithms, or training has been done or validation has been done, that is passing a quality control. Everything matters a lot when you come to the choice of the software. And of course, finally, into the confirmational sampling. This is mostly for small molecules, which will be help us to identify the target flexibility. So these all are the pitfalls that people will try to face when they want to work it with a high throughput screening technology. But however, using Discovery Studio, it can be overcome, because every year the software will be validated and the tools will be trained, and there will be a lot of data will be included each and every year, which will help us to mimic exactly before going into, when you want to test some molecules in your wet lab technique, almost 90 to 95% we can get in a certainty results when you can use this kinds of validated softwares. So how do we implement it? This is very important. So for instance, each one of us have a different perspective of the designing of a molecules or a test of molecules. So based on their requirement, we have a multiple tools, and then more than 1 or 2 tools present in Discovery Studio under the 6 different categories of protocols. Say for instance, if I want to introduce a pharmacological based molecules, then I have a set of molecules, maybe in 1 lakh of molecules. I can go for an ADME screening, that is absorption, distribution, metabolism, and excretion. This is a simple way and a mathematical calculation method using a machine learning algorithm will help us to identify using an ADME properties of the compounds. Further, when I come to know about these molecules are having good pharmacokinetic property, then I would like to know about the behavior of the particles in the in vivo system. So I will go for top set analysis. This is the most uniqueness of this tool, where you can understand to know about a classification whether that particular molecule is carcinogenic or non-carcinogenic, toxic or non-toxic, irritation, skin irritant or non-skin irritant, sensititis, all these kinds of different methods in a single mode and a multiple mode in a different gender can be analyzed using this kind of tools. And further, you can go for whether you want to screen to identify the oral bioavailability, that also can be done. And finally, into the docking technique, and then going for whether you can from here you can go for a simulation technique or you want to stop here. But of course, nowadays is becoming very mandatory. You have to go for a molecular dynamic and simulation technique using in the different tools. Here we are having a charm tool, which is already known for a longer time, about 3 decades. That tool has been integrated as a part of a discovery studio. On the other hand, people who want to explore more about the structural features of the particular compound, they can go for a ligand-based drug designing, which also includes a pharmacophore. The pharmacophores are nothing but active particle or active functional group of your molecule, which is necessary for your biological actions. So from here, you are screening your molecules. From there, also you can go for computational pharmacology. On the other hand, if you want to target screening, say for instance, this is my target A, I would like to screen at the active site or the binding site of the protein, that also can be done and be called as a structure base. These all are the simple things which I'm trying to show. Apart from that, we are also having lots of tools which will help us to understand more about the pharmacological applications in the forensic sciences. Let us move on. See, when you want to process it, you should have a certain formats to integrate it or to import it to the systems. So in the Discovery Studio, we have a multi-structure formats and then a wide range of file formats can be imported and exported. I can take a data or the molecules which I'm drawing and sketching in any chemical databases that can be exported in terms of SD or SDF format or MOL format like this, that can be utilized in the imported kind of Discovery Studio. And otherwise, you can paste and draw in different external tools, or you can insert from a particular tool using a SMILES format. These are the 1D formats, small entry input line entry system. So all these formats can be imported and exported using Discovery Studio. Once you import and export, that can be generated in terms of a library. Say for instance, how you can generate a library for screening and analyzing purpose. It could be comes from a combinatorial chemistry, where you have one molecule and different functional group, reaction position. From that way you are trying to generate, yes, of course you can use that. Or you are trying to download from an external databases, yes, we can do with that. And synthetic compounds also can be taken, or the natural product library. You have a natural product library that also can be taken, or a zinc library, Minimap bridge, or the similarity search of compounds, or a substructure of compounds, or peptide compounds. Any of these compounds can be taken and can be utilized for your screening process. And once you take the molecules, it has to be go for an ADMET process. So since if you are very interested in ADMET process, these are all different models that has been developed by using in regression methods, which is available in Discovery Studio, where it will help us to identify what is the solubility, what is the molecule have an efficiency to cross a blood-brain barrier, whether it is affecting a metabolism or not through this particular enzyme, CPY P450 family of enzyme, or it's causing hepatotoxicity, or it is having an internal, if it is getting absorption, how much absorption, whether it will go and bind to the blood proteins or not, that can be identified using a plasma protein finding. So each and every step, if you have a lax of molecules, also no problem, you can able to take those molecules and you can process. So importing will be very easy. So as I told you, Discovery Studio has 6 different protocols, and then we have different tools are present here. So when I try to open, I will go to small molecules, ADMET descriptors, load my molecules, and select all the descriptors. Once you run, you get a results along with a plot. From the plot, you can able to understand how the molecule behavior is. Each and every numbers or a numerical value represents different levels of absorption. Maybe a 3 is a good absorption, 4 is a medium, optimal. Blood-brain barrier, whether it's a 1 is a highly penetrating and 2 is a less penetrating. CPY2D metabolism, yes, it is affecting the metabolism or non-affecting the metabolism. Inhibitor means true, false means non-inhibitor. Like that, even for hepatotoxic, true means it is causing hepatotoxic, false means non-causing hepatotoxic. Absorption, good absorption. Similarly, for a plasma protein binding, like this, we have a simple letters which can help us to identify more easily for this process. And moving on, so soluble G, I can just screen within this levels 3 and 4, and then blood-brain barrier, and further into CPY2D6 family, and you can check only for an false rather than a true. False means non-inhibitor, true means inhibitor. And then moving on into the CPY2D50, which is for metabolism. You can predict the metabolism activity of this particular ligands. So once they keep on moving, you can see there is a variations in this dots that you're trying to see over here. That means that only those molecules are trying to obey the particular properties. Other molecules are not obeying the property. That is what we are trying to understand from you. And moving down, we have an hepatotoxicity properties that also can be predicted. And then depth into an absorption. And then moving on to the plasma protein binding. So like this, these kinds of analysis seems to be very easy. And then we can go and can screen for any number of molecules. It is not limited. So initially I started with the 25,000 molecules. And then finally I end up with the 46 molecules, which is obeying all the adiabatic properties. And finally, you can check it you can check for TopCat prediction using to measure the toxicity of the compound using a computer assisted technology. It basically uses classification models and regression models. The classification models that help us to identify whether it is carcinogen or non-carcinogen or mutagen, non-mutagen, which is built by using the Bayesian analysis technique, a kind of a machine learning algorithm. And similarly, in a regression model, you can be going for a partial least square, and then that will help us to identify the intermediates and screening of the compounds. So you can access for all the pharmaceutical, commercial, industrial, agriculture products for a potential safety concerns. Also, we have a set dosage range for animal assays. So it is also very easy. You have to select the compounds and you have to select the model. All these models, FDA models or food drug approval models, and NTPs, natural toxicity models. So we have a different models you can select, and you can move like this, and you can process. And then the results which shows whether it's a mutagen or non-mutagen will help us to identify by hitting with the different databases having a similar like molecules. And set dosage, which is a uniqueness of this tool, where you can identify if you give the compound, you want to see the TD50, LD50, it will predict and give you the results. And even we have environmental models also will be there. Even you can predict the TD50 values also. And then once you get those ligands, that can be taken for a docking analysis, whether it's interacting or not, that can be analyzed. So moving into this, we call it a structure-based drug designing. For this, we have that protein, and we have to define it and set ligand, and then you have to go for which docking algorithm you have to choose, and then analyzing your results. So this is an actual workflow we try to follow. First, in a protein preparation, the central assumption, good incubators must possess the most chemical complementarity as well as structured complementarity, which should include shape, size, charge, and polar interactions. And once you prepare your protein, download your protein, you have to prepare your protein. Make sure everything seems to be fine, exactly like in vivo systems. And then prepare protein and then go for binding site analysis. So we have to identify the way the binding site. It can be identified using various technology. Here we are using site-search approach and fat filling algorithm to identify the protein shape. And once you identify the binding site, you can go for docking technique. You can choose the different docking algorithms. I'm trying to explain you over here for an CDocker. It is basically a molecular dynamic simulation based docking and includes grid based methods. And just you have to double click and open input to a receptor and then ligand. That runs the protocol perfectly and you can get the results. So this is how we'll be getting results. The minus the C Docker energy value, the best the energy values are. And then you can see how the interactions are happening to understand the atomic level interactions. So atomic level interactions are measured always in terms of an angstroms. And you will get both the interaction type, whether it is having favorable, unfavorable. This is very sensitive tool to help us to understand more prominent results. And analyzing, you get automatically a tab will be added to your results, which help us to identify which atom of your ligand or which atom of your receptor is having interaction, with how much distance, category, and type of bonding. So from here, once you identify the best molecules, you can move forward a molecular dynamic and simulation also. So this is an overview about the Discovery Studio software and its importance and applications in the forensic. If you have any queries, we can always discuss. Thank you, everyone.
Yeah, thank you. Dr. Shivani ma'am, any queries, ma'am, for all of 3 products?
Sir, this is Dr. Arushi. Dr. Shivani
Yes.
ma'am will be joining us because I think they're facing a little bit of an internet issue there. But indeed, it was a very, very informative lecture for us, and thank you so much for such an interactive session, as well as giving us live demos to the 3D scanner. Thank you so much. I think our students would be having certain doubts, but we can gather the questions together and maybe then mail it to you. Because they are as we are going live on Facebook, they will be putting on their questions there.
Okay. No issues, ma'am.
Thank you so much, sir, and thank you so much, ma'am. It was a great session, and I'm gonna get back to you as early as possible.
Thank you. Yeah.
Thank you, ma'am. Thank you so much.